Everybody, it's my really great pleasure to introduce two young investigators, Jonathan Fay at UNC and Seba Catrich at USC, both people I've known and worked with for many, many years. It's a special treat today to have these two really outstanding folks present their data, which both of what they're going to present in part are currently in press in separate papers in Nature. So you get a sneak peek of some really, really interesting stuff that's going to be coming out hopefully in the next month. And in both cases, these take advantage of really disruptive technologies that have had a huge impact on our research. Of course, Jonathan is going to talk about cryo-EM, which I think many of you are aware of. And then Seba is going to talk about a brand new virtual screening approach his group has invented, which allows for gigascale virtual screening. And both of these groups are working with us currently, looking at orphan GPCRs, and likely both will be part of set-asides for the coming year. So without further ado, I'd like to introduce Jonathan Fay, who's currently an assistant professor at UNC in biochemistry. Go ahead, Jonathan.
Thank you, Brian. Everyone see that?
Yes.
Great. So thank you again. Thank you for the invitation to speak here today. I'm assistant professor at UNC, and I'm excited to share with you some of our work on an orphan GPCR involved in itch. So today I'm going to talk about members of the MAS-related G protein coupled receptor family. This family has members in the Orphan GPCR family, shown here. Let's get the laser pointer. So this is a nice graphic from the GPCR database, where David Gloriam is the chair. And you can see a number of GPCRs, about 100 or so. Over here in this area are some of the MAS-related GPCRs. And what this shows is wherever there's a red dot, that's a structure of an orphan GPCR that's been solved. So out of these 100 or so, it's not very many that have been elucidated to date. And as you know, things that are well studied have usually a lot of chemical matter associated with them, and things that are less well studied do not have this chemical matter associated with them. So an important goal, of course, is to identify structures and drugs that bind to this and shed light to this dark. So a different cladogram over here showing some of these MAS-related GPCRs, specifically focusing in on this X clade. This is a primate-exclusive orphan GPCR family, and they're expressed in sensory neurons and mast cells. And today I'll talk specifically about this guy, MRGPRX4, or just X4 for short. It's a bile acid receptor involved in the sensation of itch. Now, it's been an orphan receptor for a long time, but recently it's been shown that bile acids can bind to MRGPRX4. So the Dong lab at Johns Hopkins and Yulong Li lab in Peking showed that X4 is a bile acid receptor. So to contextualize this, you can think about this as like bile acids accumulate in the liver, get into the circulatory system, reach the skin, where DRG neurons have projections into the skin that contain these MRGPRX4 receptors. These bile acids bind to it, activate it, and the other projections go to the dorsal horn over here in the spinal cord, and then we get some sensation of itch. So one can imagine that pharmacological manipulation of X4 could be a promising strategy for alleviating itch for those who suffer with liver diseases such as cholestatic pruritus. So our first goal was to make a more selective and potent ligand. So first I'll go into a little background. So around 2015, Dr. Roth's lab came out with this presto tango paper. And so that's shown here. It's a platform for interrogation of the druggable genome. And so here's like a generic GPCR, and it's fused to a vasopressin tail with a protease site and a transcription factor. Agonist activation leads to recruitment of arrestin with, in this case, it's fused to a protease, which allows for this transcription factor to be released and go into the nucleus, and in this case, activate a luciferase reporter gene. So this is all done with arrestin, and as you know, GPCRs can couple to multiple different G proteins, so this is basically a G protein agnostic assay to screen. We can basically set this up as a cassette to screen many different GPCRs. And so that's what Dr. Wes Cruz did. He screened 91 GPCRs against 446 drugs. And an interesting hit was this compound here, nateglinide, shown here as the dose response, which binds to and activates MRGPRX4. So nateglinide has around a 5 micromolar potency at X4 in a calcium flipper assay. However, nateglinide also has an off-target effect. These glinides bind to KATP. KATP is the ATP-sensitive potassium channel in beta cells that are found in the pancreas. And these act as a metabolic sensor, and they're coupled to insulin secretion, which, reduces blood glucose. So you want to develop compounds that are selective for X4, but do not have these off-target effects. So going back to some of the earlier literature, this Shinkai et al. Paper from 1989, they found that this methyl ethyl cyclohexyl group here, when replaced with a cycloheptyl group, led to compounds that did not reduce blood glucose. So Dr. Kate Lan-Su in Brian Roth's lab, when she was getting her PhD thesis, synthesized this compound and found it bound to X4. And so that led us to a series of analog screenings, specifically looking at bulky substituents in this area. And it was eventually found that this added mantle group with these methyl substitutions here was able to bind to MRGPRX4. I'm going to refer to this compound as MS, and it bound with about 100 nanomolar affinity. And I should also point out that it has about a 50-fold selectivity towards X4 over KATP as assessed by Lily Jane's lab at UCSF. So now we have a more potent and more selective ligand. And so next we wanted to see and solve the structure of this new ligand bound to X4. And so this work was a large part carried out by Dr. Chan Kao, shown here on a late night together, making some grids. So Chan purified X4 from insect cells, combined with the mini GQ heterotrimer. And we looked at these grids in our Talos Arctica at UNC. So here's a grid, and you can see it's underneath the microscope. And here's an example micrograph. So I recorded about 4,500 micrographs of this X4 mini GQ MS heterotrimeric complex. You can see this is beautiful. And this is, a representative micrograph, but they're all like this. There's thousands that are just this beautiful. It's a really great data set. You can see these are like a beautiful monolayer, and the GPCRs are just packed in there. And I should point out, we've done, I think, around 40 or so GPCR complexes to date. And this one was one of the harder ones for us to do. We had a lot of difficulties that took some time to solve, including even some ligand-free states. But we were finally able to optimize the sample, and this is just a fantastic data set. So you can see here some of the class averages. They're fantastic. You have clear secondary structure. The overall map went to a nominal resolution of around 2.6 angstroms. And here you can see the receptor in aqua sits in this detergent micelle coupled to the mini GQ heterotrimeric G protein stabilized by this Fab. And you can see the resolution is really good. And it's sufficient to model and place many of the side chains. Now, cryo-EM combined with single particle analysis is just a powerful technique. And what I really like about it is it's an ensemble measurement, right? You're taking hundreds of thousands to millions of particles and creating this average state. And one of the exciting features in the CryoSpark package is this 3D variability analysis, which uses an expectation maximization algorithm to explore states within the ensemble. And this can provide useful information to understand this, this flexibility or dynamics of the protein in the ensemble. And I believe that understanding these states could be useful to discover new molecules that sort of limit or enhance this conformational freedom. And maybe we can design drugs that manipulate these kind of movements. So this 3D variability can be done in a cluster type of analysis or this rolling window where the particles are sort of sorted along this latent space variability dimension and then split into overlapping subsets along this variability dimension. So you can see here in the map, it's sort of twisting. And if I dock 20 or so intermediate models into each of these conformations, you can really see the receptor twists in this extracellular region about the alpha-5 helix of the G protein, and there's a little bit of variability in the gamma subunit. But the rest of the variability is quite low. In fact, when I just overlay all of these intermediate conformations on top of each other, you can see that most of this variation is in transmembrane helices 1, 2, and 7, and just in this extracellular region. On the intercellular region and the G protein itself seems to be pretty stable. Then you can see this a little bit more clearly in a top-down view. Again, we see this variability of 1, 2, and 7. And this region back here comprised of the extracellular regions of transmembranes 3 through 6 all seems to be very stable. And behind the MS compound is this tryptophan, 158. It appears almost invariant, and it's sort of, you can think of it as the center point or this pivot point for this extracellular pendulum, this twisting of the receptor. So now you can see this tryptophan residue behind this added mantle group of the MS compound a little bit more clearly. And in addition, I've got 5 residues within 5 angstroms of the ligand binding pose depicted here. Now, due to the variability of the receptor in the complex, I had to perform some local refinement of the receptor-only region, which was critical in help pulling out a clearer density for some of these more variable areas. And that allowed us to unambiguously model the ligand and surrounding residues. We used glide-EM in the Schrodinger package to help validate the ligand binding pose. This uses EM density as well as the protein model to dock and score binding poses. And you can see the top three hits here in different shades of blue. They bind extremely well and agree extremely well with our ligand binding pose. And so to take this a step further, we sought to further corroborate our ligand binding pose. And this was done with a mutational analysis. We used BRET2 or TruePath, which was developed in the Roth lab. And so alanine substitutions were created, specifically this tryptophan 158, which appears to be critical because when mutated, you can see that we have a reduction in potency and efficacy of the agonist-stimulated G protein heterotrimeric dissociation. And so some of these other aromatic residues, when mutated, also reduce this potency. And some of these charged residues on transmembranes 2 and 3 also reduce agonist potency. So this really is nice and helps corroborate our ligand binding pose. Now it's interesting is overall, you can see the binding pocket is pretty shallow and very electropositive. So this might help explain why some of these negatively charged bile acids I talked about earlier, how that might help them bind to this receptor. And of course, this variability in this region of 2, 1, and 7 in the extracellular region might allow for a greater diversity of binders. So here's a cutaway side view showing this shallow binding pocket. Here's the ligand in orange. See how it kind of sits in there. And this is a pretty interesting receptor because compared to other GPCR or compared to many other GPCRs, they all seem to cluster, the ligands seem to cluster around this highly conserved tryptophan in transmembrane helix 6, part of the CWXP motif. And you can see this MS compound is far away from this region. And you may recall from the variability analysis, this region of transmembrane helices 3, 4, 5, and 6 are very stable in the ensemble. In fact, have many hydrophobic contacts in this region. So I just show you again here in another way. So here's the side view, the ligand's in orange. Here's this tryptophan 158, and its position is in part stabilized by this extracellular loop 2 disulfide. And in yellow here is this CWXP motif. In X4, the tryptophan is a glycine, so we can't see anything there. And I should also point out that above this CWXP motif, this extracellular region of transmembrane helix 6 is brought in a little bit further. The whole pocket is constricted and bound tighter. There's some kind of contraction of the transmembrane helical bundle that you don't see in most other soluble ligand binding GPCRs. So over here is this PIF microswitch, which in X4, we lack this proline in transmembrane helix 5. So it's a little bit different, and there's some slight rearrangements there. And then down a little bit further on transmembrane helix 3, we have the semi-conserved dry motif, and of course the MPXXY motif on 7. So this suggests that interaction with the G proteins alpha 5 N terminus is similar, although I haven't shown that, and I really wanted to focus in on this difference here, this sort of descending down from the ligand itself all the way to this microswitch is a series of aromatic and hydrophobic residues, which would normally, occupy this hollow space that you often see in other soluble ligand binding GPCRs. So if we compare this to the structure of 5H2A bound to INBO, you can see that this soluble ligand is buried deep in the pocket, in contrast to X4. And you can see that this space is occupied by these cyan-colored aromatic and hydrophobic ligand or residues instead. And so overall, this suggests, an atypical activation mechanism, where you can imagine that the way that you interact with these microswitches may be for X4 more indirect versus this more direct interaction that you see in other GPCRs. So perhaps understanding ligand activation mechanism in greater detail could help improve further drug discovery efforts. So just to summarize, the presto tango G protein agnostic approach allowed for the identification of a lead compound. It was further refined to enhance binding and mitigate off-target effects. And now we're at this really amazing point. We've got a lot of combined efforts and collaborations getting us to this point, and we have a structure of X4 bound to this new ligand. I'm particularly excited about the future and what new ligands might arise from this, including antagonists, which of course would be useful, as well as maybe as the ligands mature, perhaps getting buried deeper into the pocket, or I hope we can learn about how the protein transitions from active to inactive states. As I mentioned in the previous slide, it has this atypical mode of activation in ligand binding. And I also hope that we can exploit some of these ensemble state insights to help add additional ligand contacts to variable regions to hopefully create new drugs that sort of probe how taming this conformational freedom might affect activation or inactivation. So this work is to be published and as part of a larger story. So I hope, we can talk about this in the future and some of our more exciting findings. Overall, this has been exciting and a fast-moving field. And I just want to say that cryo-EM has really been a key technique for a lot of our efforts. In collaboration with Brian, to date, we've got over 40 different G protein complex states. You can see in these violin plots, the median is a little bit lower than the median for many of the GPCRs solved by cryo-EM in the electron by cryo-EM through GPCR solved by cryo-EM in the PDB database. And it's a really exciting and fast-moving time. And in part, some of the success, aside from the advances in protein purification of GPCR complexes, some of the success we've had has been due to, in large part, the cryo-EM core at UNC, run by Dr. Josh Strauss and a talented technician, Jared Peck. We can resolve almost routinely 1.8 angstrom structures of apiferidin. And we just submitted a manuscript describing sort of our best practices to do high resolution, high speed data collection. And so with that, I'd like to thank you. And I'd like to thank everyone who's worked on this project. This has been a great project, exciting with many labs. It's been an honor and a great pleasure to work with Dr. Roth, as well as so many fantastic scientists. I'd especially like to mention Chan Cao, whose work and efforts were critical to the success of this project. And with that, I'd like to thank you again for your time.